I first want to introduce Dr. Rochelle Long. She's the acting director of the Pharmacological and Physiologic Science branch in the National Institute of General Medicine Sciences, NIGMS, Division of Pharmacology, Physiology, and Biological Chemistry. She's also our program director for the Pharmacogenomics Research Network and Knowledge Base, and it's really Rochelle's leadership that started the Pharmacogenomics Research Network about 16 years ago. The Pharmacogenomics Research Network started in the year 2000 and has been very active every year since then. And we really owe a lot of the progress, I think, that's been made in pharmacogenomics and collaborative research to Dr. Long's stewardship. So welcome, Rochelle.
Thank you, Mary. And we might have gone right past my disclosure slide, but I have none. So I work at the National Institute of Health, and I specifically work at the National Institute of General Medical Sciences. And this slide is intended to show you that I work at one of 27 different institutes and centers at the NIH. It's a pretty big place. Our mission, as of recently, 2013, is to promote fundamental research on living systems to lay the foundation for advances in disease diagnosis, treatment, and prevention. We also want to enable the development of the best trained, most innovative, diverse, and productive biomedical workforce possible. This is in our strategic plan, which is recently published, and is also the vision of our director, John Lorsch. Now, we've changed quite a bit since 2000 when the network started, as Mary described. Back then, there was a lot of money to start new, big, complex projects. The PGRN got started then. But times have changed, budgets are tighter, and our institute director's vision is a little bit different. We're a little more mean and lean now. So a couple years ago, with that changed context, we had to change for the future based upon the past successes in pharmacogenomics. The PGRN was great. It was at its heyday in the third iteration. We funded about $36 million per year. We had 14 large cooperative agreements. We had 7 network resources. We had a lot of things going on. A lot of activities were started, and I truly think the group worked together to become bigger than and better than the sum of its parts. We also have PharmGKB, the knowledge base, and together the network and its partners worked and provided a lot of information that was in the FDA pharmacogenomic biomarkers for drug labeling table. A lot of discoveries were being made by pharmacogenomics and PGRN-supported scientists and by others as well. Another large project started by the group was a partnership with the Riken Institute in Japan. This alliance led to this sequencing of many different samples, and really, the PGRN was great at brokering partnerships and was a model for activities starting around the world. Now, at our council a couple years ago, it was decided that the PGRN needed to transition to the future based upon the science, but also, and this is of course unique to us, the interests of other NIH institutes and initiatives. Another groundbreaking piece in the PGRN was working on the PGRN sequencing panel. There's a version 1, 2, and 3 of this approach, and it was used in the eMERGE network, an NHGRI-supported project. The Clinical Pharmacogenetics Implementation Consortium, which we'll hear more about today, was developing guidelines that were also being used in large projects like ClinGen and the GTR, Genetic Testing Registry, and truly making an impact beyond the network. And as you all know, the precision medicine seeds were being sown. It was announced a little over a year ago, was a big project, and sure enough, what's going to be first to be implemented there but pharmacogenomics discoveries. So where are we now? In 2016, the PGRN is different. It's a little lighter, it's a little leaner, it's a little meaner. Currently, we have a hub, and this is an organizational unit that helps to transition the scientific activities to everybody in the research community. It has a web page, pgrn.org. If you go there, you can find about our upcoming events. You can read what's in the news. You can tweet. You can probably read things there right now. It's encouraged during this meeting. You can also read, apply to join. Now anybody can be a member. This is a big difference. It was a great club, but now it's open to all. In 2016, there are a couple of awards we still have made. We have centers, which set paradigms for approaches to pharmacogenomics research. You're going to hear about some of them today with examples in leukemia and adverse drug events and statin therapies. We also have a program announcement for resources. We have two you're going to hear about today, the Functionalization of Variants Project and also CPIC. And that program announcement remains open. If you're working in an area where you actually are doing enabling activities, but it's not quite a research grant, you can possibly apply for that initiative. And R01 applications are always sought. I spent time in the last year going over the review of pharmacogenomics at the different study sections. Really, it's a hot field. Everybody wants to fund it. If you write good applications, you don't need a special initiative. So from the current PGRN site, and I anticipate going on at least for 5 years, the mission is to catalyze and lead research in precision medicine for the discovery and translation of genomic variation, influencing therapeutic and adverse drug events. But at this time, the PGRN is going to be a host for various activities. You're going to hear about them today. Again, I encourage you to join, pay attention to the hub, get your research started, and be a part of it. There are at least 6 ways we hope this will be accomplished: through producing research advances, which we always want to do in NIH, but also by thinking about sharing access to resources, partnering with other initiatives, implementing in an exemplary way, communicating and catalyzing interactions, and providing leadership for the field. So watch for the PGRN at future meetings. It's at ASCPT this year. It's going to be at ASCPT next year. We thank our meeting hosts for so warmly embracing the PGRN. We'll also be at the American Society of Human Genetics meetings. I look forward to seeing you there. And with that, I'd be happy to take any questions.